In this video, we will discuss about in vivo sialylation process to suppress inflammation. In the previous video, we have discussed that with the increase in the sialations in the carbohydrate structures, then it will cause the suppression of the inflammations in the mouse model. In this video, we will discuss the pathways that cause this increase in the sialations to suppress inflammations. Previous literature has shown that with increase in the sialations in the carbohydrate structures, it will it will interact with this sign R1 or DC sign plus, and after that if these two things are present, that is increasing the sialic acids are present and this sign R1 and DC sign plus is present, then it will cause this increase in the FcR2b receptors, and this increase in FcR2b receptors is the will cause anti-inflammations, or it acts as a inhibitory against the inflammations and then suppress the inflammation response. So this is in brief how the anti-inflammatory response occurs through the increase in the FcR2b receptors that has an inhibitory feature against the inflammations. So they need to check these pathways whether in their models similar pathways has been followed or not. So first they did is they knocked down this inhibitory FcR2b from the mouse models, and then check the clinical score of the inflammations. And what they find out is that there is no decrease in the inflammations compared to the control, that is wild type, when they use IVIG or B4STF6. This is the combinations of two different engineered antibodies which contains two different glycosyltransferase enzymes. You can refer to our previous videos to understand how they have engineered their antibodies and validated their antibodies. So in the panel A, when this FcR2b has been knocked out, we do not see any decrease in the inflammations, and same things are quantified in this panel B for the 6th day, that there is no decrease in the inflammations at the 6th day. And when they knock down this ST6, in this case also they do not find any change in the inhibitory response. The panel D is the quantifications at the day 6. Another important thing in this pathway is this sign R1, and in the absence of sign R1 they do not see any decrease in the inhibitory signals. However, when they use this HTC sign plus, they see that there is a decrease in the inflammation signals when they use this IVIG or their co-culture their engineered antibody, the B4, that they called them as B4ST6FC. They see that decrease in the clinical score or decrease in the inflammations, and the same things are quantified in this panel G, where we can see that when the sign R1 is absent, they do not see any decrease in the clinical score or the inflammations. However, when they use this HGC sign, they see that decrease in the inflammations when they use IVIG or B4ST6. So one of these things are present as per the literature that are published before this paper. So they tested it for this HGC sign positive samples, and this STT6, this plays a role in the final step where it will help in the overexpression of this FcR2b receptors to cause anti-inflammation response. And so from this experimental results, they have pointed out that the in vivo sialations follows the same process that has been previously published, that it will interact with this sign R1 or DC sign plus, and thereby and thereby cause or make the receptors towards FcR2b, so that it can ligate to this FcR2b, and this interactions with the FcR2b will cause anti-inhibitory response, and simultaneously this ST6 also helps in the overexpression of this FcR2b on the cell surface, that will further decrease, or that will further cause the inhibitory response against the inflammations. So this is the mechanisms that occurs when there is an increase in the sialations on the carbohydrate structures that will cause this suppression of the inflammations. I hope you understood how they have validated their models to show that their silation process also follows the same pathways that has been presented in the previous literature. If you need to understand more about their experimental methods or their results in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.